we have our keynote talk by a very distinguished speaker, but I'm not going to introduce him. I'm going to ask two of my colleagues to come up and introduce him because they know him far better than I do. Please help me welcome Art Lander and Ann Kaloff.
All right. Thank you, Mike. And thank you for organizing this really interesting and informative meeting. It's been a lot of fun being here during the day. And we're especially grateful to have the opportunity to introduce Ralph Greenspan.
Now, Arthur and I both met Ralph at UC San Francisco when we were neuroscience graduate students. Some of you may be surprised to know that I was a neuroscience graduate student. And Ralph had come there to UCSF to do a postdoc. Now, UCSF, as you may know, has always been an unusual place, and the 1980s was an especially strange time, but Ralph fit right in.
So, as you can see here, this was a time when UCSF had yet to become the richest campus in the UC system. In fact, we were so poor that the graduate students couldn't afford funds to buy research animals, so we had to resort to dressing up as animals and doing brain research on each other, which may explain some of the lasting effects that you see to this day in some of us.
Now, these deprivations incensed Ralph, as you can see here from his demeanor, and led to frequent outbursts of aberrant behaviors, including violin playing, pastry making, wine drinking, and most importantly, secret shadow lab meetings that we had at French cafes. And it was at these meetings that we really got to know Ralph very well.
So we learned that Ralph had come to UCSF not to do one postdoc, but actually 5. So he had support from Helen Hay Whitney, a McKnight Fellowship, a Sloan Fellowship, a Searle Fellowship, and a Klingenstein Fellowship. So basically, taking a page from Mel Brooks's movie, The Producers, Ralph had convinced 5 different funding agencies that each one owned 100% of him.
This also meant that Ralph could keep each of 3 different laboratory heads continually guessing as to why he wasn't in their lab on any particular day of the week.
So this was a revolutionary career move, but we soon learned that Ralph was no stranger to revolutionary moves, having been somewhat of a campus radical as an undergraduate and graduate student at Brandeis.
At this time when Ralph was at Brandeis was in the 1970s. It was a time of environmental crisis, a time of Russian meddling, and a time of presidential impeachment. Not at all like today. Anyway, Ralph supported his growing pastry-making addiction by writing articles for Revolution News, a student newspaper so radical that all records of it have been completely scrubbed from the Brandeis servers. It can no longer be found.
And when he wasn't revolting, he went to science labs looking for things that were revolting. And it wasn't a long time before he found something that was truly revolting.
And that would be, of course, the fly.
Now, the fly, in case you're not certain, is the one on the right. The one on the left is Jeff Hall, who was Ralph's PhD advisor. So Jeff had the idea that you could use fruit fly genetics to learn something about the neural basis of behavior. This was an idea that was not particularly popular at the time, and which may explain Ralph's attraction for it.
Needless to say, it was a defining moment in Ralph's career, and it wasn't such a bad thing for Jeff either, who received the 2017 Nobel Prize for his work in the area.
But whereas Jeff went on to focus on things like courtship and circadian rhythms, Ralph wanted to focus on the more perplexing behaviors that he observed while a postdoc at UCSF.
Now, even back then, in the 1980s, UCSF was a place with many Howard Hughes investigators, and Ralph observed that when more than one of these investigators was present at the same faculty meeting, certain behaviors could reliably occur.
And Ralph found out that he could reproduce the same behaviors in fruit flies. Now, this is, of course, something that comes as no surprise to any of us who have ever chaired a department.
Ralph also noticed, however, that the second most common behavior observed in faculty meetings after aggression was this one. And he could get flies to do this too.
So at first, he induced sleep in fruit flies by forcing them to listen to online cybersecurity training videos. Later, however, he realized it was much faster to just use drugs.
His colleagues, however, were concerned about the ethics of drugging unsuspecting laboratory subjects and branded him a fly pusher.
So Ralph took those words to heart and used the situation to launch his first autobiography. This was a tell-all narrative of the goings-on in the dark underbelly of behavioral research on organisms that happen to have a dark underbelly. That was in 1996, by which time he had already held faculty positions at Princeton, the Roche Institute of Molecular Biology, and NYU.
So shortly after these peripatetic professorships that Ralph engaged in on the East Coast, he sailed off to California. First, he was at the Neurosciences Institute in La Jolla, and since 2004, he's been Associate Director of the Kavli Institute for Brain and Mind at UC San Diego.
Ralph's research accomplishments have been marked not just by a devotion to neurogenetics, but by a willingness to take on some of the most complex and difficult issues both in neuroscience and in genetics, proving himself to be a true systems biologist before there were systems biologists. His scientific writings are characterized by a singular combination of intelligence and playful wit, and he's also weighed in on a variety of philosophical topics, including reductionism, determinism, and morality. He's recognized as one of the architects of the NIH Brain Initiative that we heard about this morning, and also the Cal Brain Initiative that he's going to talk to us about today.
Now, Ralph clearly has a lot to say today, and we want to let him get on with it, but we can't leave the stage without pointing out something that everyone who's ever worked with Ralph knows, which is his amazing dedication to teaching and to mentoring. And that's certainly benefited both of us personally when we were starting our own careers, whether it was advice on how to give a scientific talk, how to write a paper, or just how to roll out pastry dough correctly. Ralph was there for us then and has been there for us ever since, and we're very happy to have him both as a friend and a colleague. So welcome, Ralph.
Welcome, Ralph Greenspan.
Well, thank you for that stirring introduction. And as Jeff Hall once said after I had given him a 20-minute introduction, in the few minutes that remain. So as Dave mentioned, I want to tell you about the program CalBrain that I am co-director of and was co-founder of. And to do that, I need to tell you about the origins of it, for those of you who may not be familiar with the story. The reason for existence of our initial proposing of the idea of the Brain Initiative came from knowing that the limitations in neuroscience were because technologically it's extremely difficult to be able to see what's going on in the brain comprehensively enough and at a fast enough time scale to be able to analyze the things that we who do neuroscience all realize are the essence of the way the brain does things. And the existing technologies are crude. Neurology exams are the crudest of all. CT scans are low resolution. MRI, not high resolution. EEG, real time, but extremely low resolution. And cognitive and neuropsychological testing, diagnostic, but not informative about mechanism. And the current technologies that exist are, as I just mentioned, the EEG, MRI, and MEG is another one as well, that has the advantage of being real time and broad in the brain, but not as comprehensive as MRI. So I had the good fortune to be invited to a meeting that the Kavli Foundation was sponsoring in England in 2011 at this beautiful Georgian mansion that the Royal Society has taken over and made into their meeting center. And at this meeting, they had invited a whole bunch of us who are, on the one hand, either neuroscientists or on the other, nanoscientists, because the Kavli Institute, the Kavli Foundation and the beginnings of its institutes were interested in these two areas as two of the frontiers that they said were most in need of support for breakthrough findings. So I went to this meeting, and one evening during the meeting, as we adjourned to go off, so it was an interesting meeting in the sense that it was meant to get nanoscientists talking to neuroscientists, but that occurred only to a very limited extent because basically we didn't understand what they were saying and they didn't understand what we were saying. But nonetheless, there was a group of 6 of us who were excited by the idea of it, especially when at the end of the last discussion before we broke for dinner, Rafa Just from Columbia, who was one of the group in that gang of 6, he had stood up and said, well, what I think we really need to do is figure out a way of recording from every cell in the brain, every action potential in the brain as it's occurring. And so that was sort of the impetus for a discussion, which we had. Oh, and I should say that the spark for him to get up and say that was George Church, the very well-known pioneer in molecular biology, who was present and who was asked for his comments at the end of the conference. And his comment was, well, I've heard people talk about what they can do, but nobody said what they really want to do, even if you don't know how to do it right at the moment. So with that, Rafa got up and said, we need to be able to record every spike in the brain as it's occurring. And I would say the room went pretty much 50, majority of people in the room thought it was absurd and impossible and therefore not even worth discussing. But as I mentioned, there was a group of 6 of us, one of whom was George Church, who was a molecular biologist, one of whom was Myung Chun, who was an officer of the Kavli Foundation, and the other ones of which were nanoscientists and neuroscientists. I'll show you who they were in a moment. But after this meeting, we went back to our respective labs, and by email, Myung Chun from the Kavli Foundation had suggested that she had had a conversation with Tom Khalil of the White House Office of Science and Technology Policy, back when there was one. And he had been very excited. He had said that he wanted to do a grand challenge in neuroscience. And did we want to think about that? And so we, by email, started exchanging thoughts with each other about how to pursue Rafa's idea of recording from every cell in the brain at the same time as it's occurring. And out of that came a white paper in October that we submitted to him in the Office of Science and Technology Policy. And to our amazement, they were interested in it. And that started a series of meetings and discussions that went on for pretty much the next year and a half that roped in a whole bunch of other neuroscientists, nanoscientists, physicists, mathematicians, very multidisciplinary, and finally resulted in, as well as the head of NIMH at the time, Tom Insel, and Story Landis, the head of NIGM, NINDS. They were both very excited about it. And they went home to their institutes to tell them about this and said that they should be on board and start programs to be able to support this. So this was the original gang of 6. Included, as I said, included Myung, George, me, Michael Rukas, a nanoscientist from Caltech, and Rafa Just from Columbia. And we got together, it was mainly under Rafa's leadership, to pen an article for Neuron proposing this concept and talking about not so much how you would go about doing it, but the rationale for doing it. And saying that this was therefore needed to be a large-scale research effort to rope in all the relevant disciplines. And then over the course of the next month, we expanded the group and invited in more nanoscientists, Paul McEwen of Cornell, Paul Weiss from UCLA, Terry Sinowski from Salk Institute, computational neuroscientist, John Donahue from Brown, who was studying human motor control prostheses, and Carl Deisseroth from Stanford, who is developing all kinds of new, neat technologies for seeing what's going on and doing things to the brain. And out of this, we developed the idea that there were tool-building goals to measure the activity of every neuron in complete brain circuits, to be able to manipulate the activity of them so as to test their function, and then to analyze computationally what they're doing so they can be modeled. And this would have to start in model organisms long before it could be done in humans. We sent this in to the Office of Science and Technology Policy. They held meetings over the course of 2012, and in February of 2013, when Obama gave his State of the Union address, he said,
"Scientists are mapping the human brain." At which point I was sitting in my office in the early evening listening to it, and I nearly fell off my chair. Because I was talking about our proposal, which was indeed the case. We got emails later that a couple hours later from Tom Camille saying our project had been chosen as one of the great challenges.
So then in April, on Good Friday, we all got emails saying, show up at the White House next Tuesday for a meeting on research initiatives regarding the brain. So we all dutifully showed up, and it was the president and the head of NIH at the time, Francis Collins, who introduced Obama as the neuroscientist in chief of the country, and they announced the idea of this joint program that became the Brain Initiative, that began with NIH, but quickly roped in a bunch of other agencies as well. And it was an initial $100 million allocation for things that would involve DARPA, NIH, NSF initially, and then subsequently expanded to
other government program research mechanisms. And
and it was an important component was going to be having NIH ethics be incorporated into the way the program was organized and the research that it would do, because
of the potential ethical issues that would arise from being able to manipulate brain function. So this was announced, and as often happens with radical new ideas, there was a backlash of people saying, well, it's a waste of money, even if you could do it. First of all, it's impossible, but even if you could do it, what are you going to do with all those data? And George and I were both having genetics backgrounds, so old enough to remember that had been exactly the response in the a couple of decades earlier, to the idea of doing the human genome.
They said they said it's too difficult, you can never do it, and even if you could, what could you use all that data for? So that was what we pointed out to them, and then the NIH, NSF, and DARPA, and all these other agencies gradually rolled out their programs. So sometime early that summer, I got a phone call from a staff member in the office of the California Senate not the majority leader, but somebody else who sort of had an administrative role in the California Senate, saying that they had seen they had heard about what we're doing because there were all these newspaper articles that the New York Times and other papers came out with announcing the existence of the Brain Initiative. So when we said that we were going to start this California program, they decided to hold a symposium at UCSD where we invited a bunch of scientists from California, Bay Area and LA area and San Diego area mostly, to come to talk about what could be done along these lines for developing new technologies for understanding what's going on in the brain. And thus began the program which we named CalBrain, which was jointly directed by Paul Alivisatos, one of the nanoscientists involved in the original germination of the idea, and me as co-directors. And we had an initial allocation from the state that was sort of earmarked by the state Senate majority leader for us to use to seed grant the program. So we started a program of $2 million seed grants, and we were able to give out 16 of them, which involved 12 different institutions, and it was for state and private institutions in California that were doing leading research in neuroscience. In order to follow through on this at the state level beyond that first year of allocation back in 2015, we then worked with our legislative representatives at UCSD, and with them, we were able to find a sponsor to propose a bill for the California State Assembly to designate CalBrain as the official recipient for innovative research funds that were allocated as part of the Mental Health Services Act in California, which was a program to fund mental health programs and the mental health clinics in all of the counties. And there had, at the very outset of that bill, which was back in the early 2000s, they had stipulated that some portion of it should go towards innovation. And it turned out that that never happened. Instead, it got soaked up by the counties for their own budgets. But with that as our ammunition, we went ahead and they were able to pass this law, which said that we should be the official recipient for those funds to allocate them to research labs. And they didn't allocate any budget to us because the budget for the State Mental Health Services Act is decided in a decentralized fashion by the county mental health officers. So all we could get was the official designation of it, but nonetheless, we still had that initial allocation, which we used to fund a bunch of seed grants to try to address the problem that the current technologies are low resolution and slow, and that our limitations in neurology are because we know so little about what goes on in the brain. So essentially, we made the argument to direct this program towards finding the new technologies that could solve these problems for new diagnostic tools, new treatment tools, and new understanding of the fundamental mechanisms so the tools could be developed, and for things like neural prostheses. And that in order to get there, in order to be able to make these things work and be useful, we needed to know a great deal more than we did now, and we needed better tools for being able to correct such deficits, or at least to try to correct them. So out of this has come some nascent new technologies, which I'll tell you a little bit about in the rest of my talk, which are new tracers for brain imaging, including being able to detect inflammation for multiple sclerosis and TBI, and for detecting memory formation in the brain through histone deacetylase proteins. Prosthetic tools for ameliorating, for enhancing and correcting deficits in various kinds of brain function. Nanoscale biocompatible sensors of nerve cell activity, at least for animal models initially, to see if they could be made so they could work, and then see if they could be made tolerable by humans. Wireless devices for targeted therapy, and new modalities for brain imaging, such as magnetism and ultrasound. So I'll tell you what some of these research programs are doing. One of the most important and new ones is from Miriam Xiaomei at UCSF, and she's developed tracers for metabolic imaging in the brain for as a diagnostic tool and as a way of monitoring progression using hyperpolarized carbon-13 probes. And this makes use of magnetic resonance spectroscopy, which is sort of a fancy way of saying MRI, for looking at, in a non-invasive way, at the appearance, progression, and formation of these neuroinflammatory responses in the brain. And they have taken this to the point now, starting with their CalBrain seed funding, of getting very significant funding from various foundations and ultimately from NIH to pursue it. And they've become an NIH resource center for this technology. So, and this shows you an example of two of their probes. One is lactate and pyruvate in the brain, and the other is bicarbonate and pyruvate. They do a ratio imaging of the lactate to pyruvate ratio and the bicarbonate to pyruvate ratio as metabolic measures of inflammation, which make it possible to detect inflammation in the brain in something like traumatic brain injury long before any of the other imaging modalities would detect it, and much sooner after the injury, so that there is a lag period in TBI between when the individual sustains the injury and when the symptoms begin to appear, which is often 3 to 6 months. So it gives you a long time and it takes a long time to develop. So if you can detect it very shortly after the insult to the brain, there would be lots of time if you could develop therapeutic ways of approaching it to limit the extent of that inflammation. And this shows you the mouse studies that they did ahead of time, where on one side it shows you the standard MRI, and the other the MRI with the tracer. And this is the upper panels are the injured brains, lower panels are control brains. And this just shows you the ratiometric way that they were able to show by the spectroscopy the ratio between in this case, the lactate and the pyruvate. And this shows you sort of a time course of development of them post-injury and the cytological manifestations of it in the brain. And this too shows you the progression over time of it. So the idea being that you see it at week 4, which is dramatically earlier than when symptoms actually appear, and would really be an enormous boon for being able to know who was in need of therapeutic interventions. And then this shows you more of the local imaging and of the spectroscopy results. And these were so this was for multiple sclerosis in an animal model in which you induce it by inducing an autoimmune response. These are for TBI, where it was induced by injury to the mouse, and where you also see the progression of it as after the injury, so on. Another important new project, which is an imaging advance, was done by Marcelo Wood and Mike Yassa here at UC Irvine, developing a PET tracer for epigenetic modifications, which they did originally for looking at age-related memory dysfunction in the brain, but which has broader applications than that. And this involves a carbon-11 molecule tracer called carbon-11 martinostat, which detects histone deacetylase enzyme density in the brain and can be used as a marker of memory formation, because HDAC activity is one of the molecular markers for memory formation in the brain. And this is a form of memory that's significantly impaired in aging mammals, rodents and humans. This shows you in a monkey brain the demonstration that this shows that this is a way of measuring HDAC density in the brain. And they've also shown that there's a very individualized fingerprint of epigenetic modification in the brain that is characteristic of the individual, not surprisingly, given that none of our brains are absolutely identical to each other. And that it also is useful as a way of measuring progression in Alzheimer's disease. Leah Krubitzer at UC Davis has been doing some interesting studies on a tool for not just monitoring and imaging brain activity, but doing localized cooling of tissue for tissue protection during neurosurgery. And for this, she's developed a device that in animal models gets inserted into the brain and provides localized cooling for that brain region. And in this demonstration, in a reaching task for a monkey, she found that you cool the appropriate motor regions, and what you get is a sort of a reduction in the ability to do a grasping task as a consequence of that, following the cooling. And this shows you the imaging of the onset of cooling and the regional expansion of it in the first minute. And then this shows you what it looks like after you rewarm it afterwards. That it's as you would expect, it's a easily reversible phenomenon. Another promising project is in Richard Anderson's lab at Caltech, in which they've developed a device for prosthetic control of a robotic limb as part of his studies of motor function in the cortex. And in these studies, what they've done is to develop a way of, initially done with primates, taking recordings from the brain and using those recordings to drive the movements of a prosthetic limb in a kind of a feedback loop, where they would record the signals in a primate as it would do a reaching task and interpret them computationally as a way of signaling to a control limb. And this would be the way that it acquires sensory information for it, and then you could deliver that back to the brain in a kind of a feedback loop. So you could do this, they've been able to do this comprehensive mapping of the motor cortex in the brain and taking those signals and feeding them back into the brain. Miriam Shinecki at USC has done something along the same lines, but not identical, in which she has done recordings from the primate during a reaching task. As she's done it actually with sort of two parallel animals, one receiving the sensory stimuli, the other receiving the feedback from the computer recording and reintroducing of it. So this allows them to record the population activity of these neurons as they're doing this adaptive response after brief training, and using it to drive an avatar, in this case a monkey in an adjacent chamber, to carry out the same tasks. And this is just an example of the population recording that they do and the analysis of it that they make use of. Then along the lines of sort of new modalities, new recording kinds of technologies, Carlos Pereira-Cayo and his collaborators, Sebastian Karpf and Bahram Jalali, have also developed a new microscopy approach towards being able to have spectrally targeted signals in the brain, to the brain, that if you record them, if you're able to record them, can give you very sensitive detection and can be shaped. The wavelength can be shaped to target different optical receptors that you might use in the brain, so that you could have a kind of a diverse response within a given region with the appropriate probes to carry them out. And this, as in many of the CalBrain technologies, is something that is way futuristic, but that's the kind of thing that we wanted to support, because that's where the breakthroughs come from. And then there's another program from Bruce Cohen at the LBNL in Berkeley, developing activity probes with toxin molecules that have very specific binding properties in the nervous system, such as snake, spider, and scorpion toxins. So the idea is that they use quantum dots for producing signals, and that they couple the receptors to these, and the receptor conformation change then gives them a way of sort of seeing the activity of those cells. And they've developed the chemistry and the technology for producing these things. And I think that's my last slide. So I'll be happy to take any questions and answer them to my best ability, although I have to say that my depth of knowledge on some of these new technologies is not overly impressive, but I will do my best.